Good morning, everyone. Welcome to our, it's our fifth monthly British Columbia Poultry Symposium webinar. And I can't believe how the time is flying by. Thank you very much for attending. I again want to thank all the sponsors who have so generously contributed to our meeting. And I also want to up front thank Dr. Ryan Snyder for agreeing to come and speak to us on such an important topic. Happy St. Patrick's, everybody. May you have a great and lots of luck today. And I'm looking forward to becoming immune and traveling around again and seeing everyone soon. Dr. Ryan Snyder just completed his PhD at the University of Guelph under the University of Guelph Department of Pathobiology and under the guidance of Dr. John Barta. And we're really happy for Ryan, and we really appreciate his energy in this field. Currently, Ryan is employed by Grand Valley Fortifiers, who's also sponsoring part of our sponsorship group in Ontario, and he occupies the position of Poultry Production Improvement Specialist. So with that, Ryan, I'd like to give you the audience and welcome to British Columbia. Talk to you soon.
Yes, thank you so much for having me, Dr. Ritchie. And can I just get somebody to confirm you guys can see my slides, the title slide up now? Perfect. Okay, thank you, Stu. Good day to everybody. To those who are in BC, good morning, and those out here in the more eastern side of the country. And then I've understood that there's a few people joining in from the US. And as Stu said, happy St. Patrick's Day to everybody. So today I'm going to be talking a little bit about platinum brooding, which is part of a program to get your chicks off to a good start, as well as coccidiosis prevention. I'm not sharing anything that's wildly new today, but I want to just cover a lot of the basics. Maybe it is new information for some people. We will dive a little bit into the science of the cocci, and we're going to talk about preventing the disease and the different methods that we can do that. So as Dr. Ritchie mentioned, I did my PhD at the University of Guelph, and I just completed that last fall. And for the last several months, I've been working at Grand Valley Fortifiers. And just the plug for me to introduce the company, for those of you who are not familiar with us, we deal with premixes and on-farm feed manufacturing. And this is across Canada, and we do a little bit of business in the US as well. And but we're not going to be talking too much about nutrition today. We're going to be talking about cocci. So if you were to search the literature for either coccidiosis or Eimeria, these two graphs here are in reverse chronological order. So the left is 2021, and then the right is 1997. And you can see that over the last 25 years, coccidiosis and Eimeria publications have just risen. So to a point where in 2020, we had over 300 publications with the term Eimeria. Now that could be non-chicken based, but at least coccidiosis is more chicken based search. So we are almost at the point of having a publication every single day of the year. So that's pretty remarkable how much work goes into coccidiosis. And this is not surprising because coccidiosis and necrotic enteritis, which are both linked with one another, we're going to talk about that today, are ranked as some of the most important issues to veterinarians at integrators in the US. So already we can tell that this is a really hot topic, and I'm pretty sure I'm not going to be able to cover over a century's worth of information because we've been looking at coccidiosis since we started commercial production for chickens. So I'm going to do my best to kind of talk about a variety of different things. But first off, why is coccidiosis important? Why are we looking to control this disease? And we are going to talk about the disease today and how complex it is. But part of it has to do with why this is important is that we've been losing some drug effectiveness. We've had anti-coccidial drugs available to us for decades, and they are still very successful at preventing disease, but there could be some room for improvement.
A little bit of a freeze spell. I'm not sure what's going on.
Oh, Pardon, can you see my slide? Sorry, Stu, can you not see my slide?
Yeah,
Yeah, all good.
you're good.
Okay.
You're good, Ryan.
So not only are the drugs losing some effectiveness, but we are also losing some drugs that are available to us due to regulations, due to the RWA movement, raised without antibiotics or no antibiotics ever. Coccidiosis control becomes important for vaccination because we need to understand how to manage the flock in the barn properly. And these infections, not only do they lead to disease, they can also, disease of coccidiosis, but bacterial infections such as necrotic enteritis. So some of the T's towards the end of this conversation today, what are the options for the future cocci control? These are some ideas. I don't know exactly how it's going to look, but I believe that we may end up with maybe chemical anticoccidials only. We may look at a yearly rotation between medicating with these or vaccinating in the summertime. We could look at the program of a bio-shuttle where we use the vaccine and the anticoccidials within the same flock. And then we also have the natural products that are available to us. And maybe that these natural products could become more effective on their own. But I do think that we will see these nutraceuticals used in combination with either the anticoccidials or the vaccine. So I'm going to go over the different species of Eimeria that are infecting chickens. We're going to talk about how the disease is artificial. I want to go through the life cycle. This is where we're going to get into the nitty-gritty science of it, and then how we're controlling the coccidiosis inside and outside of the bird. And then I want to share some of my experiences with the Eimeria that I've had over the years. And I'm going to go quickly. Here's your warning. If I haven't been speaking quickly already, I'm probably going to be speaking quickly for the remainder of this. But that's because a lot of this stuff isn't groundbreaking. But I want to give more time to cover everything and then give us more time for questions and answers near the end. Okay, so when we're looking at coccidiosis, it's really not just one disease. We've got several different species of Eimeria. Up until a few months ago, we only had 7 or 8 described species of Eimeria, but there are now actually, there's 10 now. So just last year, there was 3 different types of Eimeria that were never described, so therefore never formally named. They were now formally named at the end of 2020. So we now have 10 or even 11 described species of chickens. And they all differ in the area of the gut that they infect. So this figure here shows the gizzard, followed by the intestinal tract. So the red area of this figure shows where the birds are infected. And then they also slightly differ in the size of the oocyst as well, but the size of oocyst is not always indicative of the species that's present. So the main species that we really are paying attention to include Eimeria acervulina, Maxima, Nicatrix, Burnetti, and Tenella. The others include Bovati, Mitis, Praecox, Hagani, and then the 3 new species. But we're not going to really focus on the rest of those. I just want to look today at these major disease-causing ones. So the one that probably almost everybody's familiar with is Eimeria acervulina. This is infecting the duodenum of the bird. And it is pathogenic that it causes disease compared to some others species such as Praecox, which doesn't cause as much of an impact on the birds, but does infect them. Acervulina is pathogenic that it causes impact, but it's not as deadly as some of the other species. So like I said, it's going to infect the duodenum. It's very easy to see and diagnose because it makes these white little plaque marks in the gut. And when I look at acervulina infections, they are very prolific. So you'll see them in vast numbers if you look at the oocyst counts or oocysts per gram. But it doesn't really hit the birds as hard as it does in other species. And what I think the consensus on acervulina is that it'll take a few points off of your feed conversion. It's not really going to show in the barns very clinically. You're not going to see the birds struggling. You'll just lose a touch of feed conversion with these infections. So there is still economic impact by having these infections, but it's not in your face as a problem as compared to some of the other species. If we look at Eimeria maxima, this is in the middle of the gut. It produces a lot of mucus and inflammation. And you can see in these photos just what I'm talking about, like that sloppy orangey red color. And this is going to be a little bit different than when we talk about Tenella, this red. But this is in the jejunum and the ileum, and it's right in the area where necrotic enteritis tends to be an issue. So whenever we look at necrotic enteritis, Eimeria maxima is the one that kind of leads into that. Nicatrix, it is highly pathogenic and very deadly, although thankfully we do not see this typically with broilers. It is more of an issue with layers and breeders, so we have to be aware of it, but I am just going to stick with broilers today. But there is the odd case where this could be an issue as well. And then Eimeria tenella. I like this photo on the left here because you can see the top of, this is the ceca pouch, and this is the top of the ceca pouch, how damaged it is at the tip versus at the base of the ceca. Like these lines that you see going up and down the ceca, that's what it should look like. Now ignore any of the red dots you see in there, but those lines are virtually gone up here. So it is easy to see tenella as well. And when you open up, you can find either a red or a white core. So this picture on the far right, you can see there's white inside, and that is just chock-a-block full of oocysts. So it's pretty remarkable and quite easy to see a tenella infection. And you don't even have to open up the birds. You'll see blood splatters on the floor and an increase in mortality. So obviously a good reason to fight these parasites and control them. So like, let's go back, because we've been working on this for so long. How have we not solved these issues? And that's in commercial production. So let's take a step back. Well, what was the disease like in in jungle fowl? And you got to think that these parasites have been working on infecting this host for millions of years. So they've come up with a way to not always kill the host in a wild setting, because you're going to have a small number of birds over large areas. So the parasites want to get into the host, replicate, and then find a way to get to the next host. We're going to talk about that in the life cycle. So when you look at a wild animal like the jungle fowl, you've got some mixed ages, populations coming and going over a very large area. You have a small number of individuals. So the level of exposure of the parasites to the host is kind of low. So in the end, you don't have clinical disease. You would just have a lot of birds with low infections. And then immunity will build up over time as well. We're going to talk about that today. But so then how does this differ? Why did this change as soon as we put them into a barn? We've got plenty of infection, but little disease here, but now we're taking all these animals and concentrating them. Okay, we've now increased the pressure of how many oocysts are in an area. And we've basically given the parasites the best environment because it is warm, it is moist. We're going to talk about the biology of the parasite later. And then we've got lots of hosts, the chickens, all in a confined area, and they are all naive. They're all young animals, and they're just ready for infection. Now, similar to the wild animal, once these chickens do become exposed, they will develop a natural immunity, and it is only species specific that they'll develop that immunity for. But we still need to get to that point and grow a chicken efficiently. And the other thing that we don't often talk about is the natural behavior for chickens, and these are poults here, but their natural behavior is to investigate their environment for food with their mouth. So maybe they're not even going to eat the shavings or the straw or the feces, but they're at least going to be pecking at it to find out if it's food. So just the natural behavior of a chicken is going to accelerate a fecal oral pathogen such as coccidia. So when we want to look at coccidiosis as a single bird, we can use the lesions as we looked at before with the different species. Now, it is going to depend on the breed of bird, the sex of the bird, the type, if it is a leghorn versus if it's a broiler. The more parasites that go in tends to cause more disease, and we'll show more about that on the life cycle. And really, if you were to give a bird some cocci and just give it once, within 7 days, that infection is clear. viruses and bacteria may continue to grow and cause problems, or the bird has to mount immune response. The parasites leave on their own after a week because that's just their life cycle, and we're going to talk about that. And when we look in the barn, you know, as a bird may only have it for 5 to 7 days, it goes from bird to bird and it just blows up across the flock. And so when are our challenges of coccidiosis in the barn? Typically, we're seeing this around 2 to 3 weeks of age in flocks that have been vaccinated for cocci. And those on the conventional program that are using anticoccidios are going to have their highest challenge about 4 to 5 weeks of age. Now, which is better? Do you want it earlier or later? Well, the problem with the vaccinated flocks is that they have the infection at the prime time for necrotic enteritis. However, on the medicated side, even if you don't get necrotic, the cocci challenge has a bigger hit to metabolism. And so what I mean by that is if you were to give the same challenge to a bird that is 3 weeks old versus 4 weeks old, there could be a bigger reduction in the overall feed efficiency of that bird on the older bird compared to the younger bird. And it kind of has to do with the animal's absorption of nutrients at this age. Okay, so let's dive into the species, the Eimeria species life cycle. So this is a little bit complicated, but we'll take it down step by step. So it starts off with the letter A here, where the host, the chicken, is going to ingest an oocyst, and the oocyst has to be infective, and we're going to get to that later. So once the oocyst is ingested, it's going to break out, okay, and now it's going to go find itself a villi. So when it's breaking out, it's going to be used by the mechanical processes in the stomach. It's going to break down and open that oocyst, and then there's going to be the enzymes from the pancreas that's going to help let the oocyst find its way to the intestinal tract and know it's in the right area. And so then in part B here, the parasite is going to just slip right into the host cell and then undergo asexual replication. So it almost acts like a bacteria here, but to a certain point. So it's going to replicate, and then it's going to say, Kate, stop. We've reached our capacity now. And this is predetermined, like there's the genetics that tell it exactly how many to produce. But at this point, that's basically starting to swell the villi, and then they're going to burst out. And now you've went from 1 to 50 parasites, and those 50 now go infect the next one. So that's what we're seeing here in C and D, that is just another cycle of this. And that's kind of why in cocci you might see these cycles happening even before the true complete life cycle is done. There's these cycles of infection. So there's these cycles of asexual replication, but then after a while, again, genetically determined, the parasite says, okay, now I need to say, I'm going to turn into a male or a female gamete. So in E, we see this one starts to become microgametes, so male gametes, or on H and F here, we say that they become macrogametes, and then it's just like sexual replication. We need to fertilize the macrogametes with the microgametes. And for every microgamete that fertilizes a macro, we end up with one oocyst. When this oocyst is now released into the intestinal tract and then flushed out and into the environment, it's first off not infective. This is referred to as unsporulated oocyst. And it can take 24 hours for this process to work, to become sporulated. And now it's ready to be ingested by the next host. So if a bird were to come along and pick up a piece of fecal material with these unsporulated oocysts, there will be no infection. Okay, so this whole outside of the bird becomes important when we're looking at vaccination strategies. So like I said, we've got a part that's inside the bird, and we've got a part that's outside the bird or inside the barn environment. And so we've already spent some time looking through this. I just want to share this video with you guys. This is what you're seeing is a sporocyst coming out of a sporozoite. Just a little single-celled parasite, but looks like a worm, and it's just hatching out of its egg. This is just how that they're packaged. We don't have to go into it too much, but there's basically 2 of those parasites in a single package, and then there's 4 of those packages in an oocyst. So each oocyst has 8 parasites in it. So that's pretty cool. It's always scary to see. In this video on the right, you can see this is where the sexual replication is happening, and then there's a lot of macrogametes trying to all fertilize microgametes trying to fertilize a macrogamete. And these are just some cool microscopic photos to help us understand kind of what's happening inside the bird. But obviously we had to take some scrapings, and this is what we're looking at here now. Okay, so when we look at what's going on in the barn, there's a few things that are required for that oocyst to develop and obviously survive. If you mess up the management of your barn at this time, you can really kill the parasites and kill the cycling, and now your birds are going to be at risk. So what's required for oocyst development? First off, sporulation is an aerobic process, so we need to have oxygen. That's pretty easy because we're not growing chickens in space, so there's going to be oxygen available to the birds. However, if you were to bury the oocyst, there may be insufficient oxygen, especially if there's some microbial activity where like all the oxygen is being used by bacteria. But then again, the birds aren't having access to that material anyways. So we need oxygen. We also need moisture. So the oocyst can actually become too dry and then crack, and then the insides are exposed to the environment, which they can't handle, because that oocyst wall is meant to be very sturdy to protect it from a lot of environmental stresses, including heat. You can put oocysts into industrial bleach for 10 minutes and they're not dead. So it's remarkable how hardy these things are. And we'll talk about that when we look at control later. So for sporulation, we're looking at the relative humidity to be closer to 50%. I know this says 30 greater than 30% reduces, but in reality, I think we're looking closer to, if you get too high, then you might have issues, but aside from sporulation. So everything above 25% relative humidity, I believe, would start to do a good job at sporulating. We also need temperature. And again, we're not raising our chickens in Antarctica. Our chickens are going to be at the appropriate temperature anyway. So this is kind of where I was saying we've made the perfect environment for these cocci to survive anyways. So it's just keeping the management in mind to allow the cocci to thrive whenever we're going to use the vaccine. We'll get into that. So these three things all come together for sporulation, but you can have the best environment for the oocyst to sporulate, but you still need to have the chickens find and re-ingest for our cycling to occur. Now, obviously, if you're on a conventional program or using medication, none of this matters, but this is mostly applicable to vaccinating. So let's look at controlling coccidiosis. And so I am not aware of any way to eradicate the disease or eradicate the parasites. And yes, there are probably products that you can use to disinfect your barn that may have influence on cocci. However, is there anybody out there who is using disinfection only and not using vaccine and not using a medication? I don't think so. Nobody's relying on disinfection only to eliminate cocci. So that's why I have an X there. But even just as simply as washing your barn, I believe, would reduce the load. So what we do have available to us, as mentioned earlier, includes live vaccination and anticoccidial medications. And so let's briefly go through those two. We'll touch first on the medications, just because we've been using them the longest. And historically, they've been extremely successful at controlling and preventing these diseases. When they came out decades ago, it was like a miracle. And that has led to us being such a successful industry, is getting rid of this parasite. And over the years, we developed a number of different programs, including shuttle programs, rotation programs, to not use the drugs too much, too quickly, in order to prevent the parasites from becoming resistant. However, we still are starting to see a bit of resistance. Now, we are concerned with the resistance, but it's not like the drugs are failing. Okay, we're not seeing drugs that go into a barn and then just all of a sudden we have outbreaks of coccidiosis. I'm not saying that. When I talk about resistance, I'm talking about just subclinical light infections that could be sneaking through, such as even A. cervulina. Like I mentioned, it may just take off a few points of feed conversion. And the other thing is, if this starts to get worse and worse, could we be seeing birds start predisposing to necrotic enteritis? When we look at the two classes of coccidiostats, we have ionophores or chemicals. So the ionophores, they have some importance to human medicine because they have antimicrobial activity. So in Canada, they're not allowed and raised without antibiotic production. Whereas on the other hand, the chemicals, they are strictly used for coccidiosis prevention only. So there's no antimicrobial activity. So they're not important in human medicine. So they are allowed in RWA production here in Canada for the time being. Does anybody use it? I'm not aware of it, but it is on the label that it is allowed as far as I'm aware. So it's been great. We've had a whole cocktail of different products brought to the market for us to use. And like I said, they can kind of fall under either ionophores or chemical anticoccidials. And then the one Maxiban fits right in the middle because it's got a bit of both in it. But not too much to talk about more on that. So let's switch over to cocci vaccination. And this is basically our main alternative to using the drugs. And we're going to use the vaccine mostly in the RWA programs or the NAE programs as it would be in the US. And the ones in Canada, for the most part, are live parasites. We do have attenuated, but they're just weakened. They're not necessarily dead, like a virus maybe, or a vector vaccine. So these are live parasites, and so they have potential to cause disease if left unchecked. But they are going to generate immunity, and this becomes important for flock management because we need the birds to get multiple exposures. I was going to mention the COVID vaccine, but let's not go down that road. Let's stick to cocci today. So we need multiple exposures for the birds to develop immunity to the cocci, and that needs to be done in the barn. Okay, so that means when we get the vaccinated birds, we're going to let the vaccine cycle through, as we were talking with the sporulation, and this is going to let the birds become immune so that at a later age they're protected from this. One important key point that we are going to dive into a bit later, the parasites that are in the vaccines were collected from the field decades ago, so they're drug sensitive. So basically, before the parasites had a chance to develop resistance, somebody had taken samples of the parasites and stored them. And then over the years out in the field, the parasites have developed resistance, but so now they're almost like two different parasites altogether, different strains, let's call them. So that's gonna be important. We're gonna talk about that later. So this is a few years old now, so I need to get this updated, but the vaccine market is quite large. There's a number of different products by different companies. As far as I know, there's only 4 companies who are selling in Canada. I'm sure there's a lot more available in the US and worldwide. So I want to talk about brooding the birds, getting them off to a start in order for the vaccine to cycle efficiently. So it starts with the birds being vaccinated at the hatchery. And this will be with the species that we deem important to the production. And when the parasites go into the bird, like I said, they're going to cause a little bit of damage, but we're going to keep the number of parasites we give to the birds very low so it doesn't harm them too much. So that's why there's only 3 little oocysts here, and this baby chick gets those 3 oocysts, and now the cocci is going to replicate in the gut. So we have a few more oocysts come out, and that's our first cycle. And again, we want the oocyst to sporulate. We want the birds to ingest it. So our second cycle, they've now picked up more because the first time they only had 3, now there's 5. So it's just the cocci is now building in the barn, and we want this. And then, you know, the birds are going to age, the cocci is going to continue to cycle, but eventually the birds are going to start to develop some immunity. And so you see this pile of oocysts that comes out at the end of the 3rd cycle is now bigger than what comes out at the end of the 4th cycle. And that's just starting to demonstrate that we should see the birds mounting immune response. And eventually, if you had the birds in the barn long enough, you would see them become sterilely immune. Now, I don't think we really see this, and I'll show some field data later that the birds are still shedding by the time they go to market at 5 to 6 weeks. So is that one bird that's still shedding, or are they all shedding a little bit? Hard to tell. But in any case, with multiple exposures is going to become more protected flock. So this is what we're trying to generate. And it really all comes down to that first week or 2 weeks that you manage your barns properly. And that'll set you up for success for the rest of the flock, especially on these vaccines. So it's about balance when we're looking at managing the barn's internal environment. So if you were to theoretically, you know, keep it extremely dry, you would kill the vaccine essentially. Like, yes, they were vaccinated, they come in, they are still vaccinated, but you're going to stop all cycling if you were to have too dry. And then this could lead to a risk of an outbreak later, because now say your birds have only had one exposure and that's only in the first week. Now we're going to fast forward to 4 weeks of age, and then somehow another cocci gets introduced. Those birds aren't ready, and it's just going to explode the infection. On the other side of relative humidity, you know, if it's raining in the barn, yeah, you probably could have good sporulation, but who wants a wet barn? And this is going to be a challenge for the risk of bacterial growth. So there's going to be a sweet spot where we can keep the sporulation of the cocci in mind, but also a nice dry environment to reduce bacterial growth and then let the birds have a more comfortable life going forward. So I kind of lean towards targeting 60% relative humidity and 30% litter moisture. So there's a number of tools you can use for this. This is just a, I believe it's a meter you would put on plywood or even concrete to just tell you the moisture content of it. Or you could use just a regular relative humidity sensor. There are other ones that have like little forks on them that you can stick into the litter. And so, you know, you want to be looking for this target. And so litter moisture, again, is different than relative humidity, but looking for litter moisture to be about 25 to 30% after 4 or 5 days. And so, as mentioned before, density does become important. And so if you were to look at what density you should be using, it could depend on what your litter moisture is. Because if you can't get your barn any wetter or to the optimum moisture, then what do, right? Like you don't really want to be going out and taking a watering can all the time. So in that case, you could just change your stocking density and keep the birds corralled in a more concentrated area for longer. So let's say we have this dry environment where it's less than 25%. You would just keep them at a half a square foot per bird for a little bit longer than if it were a wetter environment where you can start to let them out and have more access to the barn. And you know, it's all going to depend on what's happening outside of what you can produce inside. This figure shows the seasonal, I call it summer and winter, but really it's the whole year of how much we see between our seasons. And in Canada, and especially specifically Ontario, at least, all of our barns are closed up. We have a very small vent. Whereas if you were to look at poultry production down in the southern states where it gets much warmer, they have curtain-sided barns. So the airflow is going to be totally different because the outside temperature isn't as extreme. So just as an example, you could start your birds off at half a square foot with one wall here. And then after a week or so, or 2 weeks, you can then let them to the rest of the areas. That way you're not going to keep them confined too much where it gets too soupy in that one area and then let them out. You can just a gradual way of managing the barn to keep the birds condensed, I guess. So again, you could cut them in half and leave them on the side where the heaters are. But one thing that I've been more curious about trying is, and I don't have a figure for it, but rather than have, you know, a 200-foot wall or a 500-foot wall going the length of the barn, what about cutting the barn in half this way and having the chicks on one end and not on the other? And then that's a shorter wall. Yeah, you got to go around your feed equipment and your drinker equipment, but it's worth a shot if you're trying to find different ways of managing your house. So as I showed before, you start them off and then you let them over, and you can either brood them with a wall, or you can try and remove the light, the food, and the water as best you can, as you can see in this photo, and that kind of keeps the birds a bit more condensed in that area. But it's not foolproof because after 5 days, they're probably going to be all over the place anyways. So as mentioned before, not managing your cocci properly can lead to an outbreak. It's not just coccidiosis. We're also looking to prevent necrotic enteritis. So another important point of the vaccine strategy, we'll call it, is that we get application to be as uniform as possible. We don't have the chicks all lined up, you know, ready to get their vaccine when they're at the hatchery. That would be really handy, but we take what we can get. It is nice that we can vaccinate our chicks at the hatchery because you can have a high volume of birds. It's automated and they can just be put through and vaccinated quite quickly. We want the birds to ingest the vaccine, okay? It has to get to their stomach and then eventually their intestines to work. Like, we're not going to be injecting it subcutaneously or intravenously. Like, it's meant to be ingested We could try to target the eye. That's absolutely an option, and then it would end up being swallowed through some ducts. But really, it's all the same thing in the end. We're trying to get it to the gut. And so we have two different ways to apply the vaccine. It could either be a coarse spray or gel droplet, depending on the vaccine manufacturer. And it doesn't matter which one you use, both are equally successful. The question is, what are you doing as a hatchery, or what is your hatchery doing for your chicks to maximize the ingestion? Because when the birds get this vaccine, especially on the gel droplet and the coarse spray, they need time to ingest it. It gets sprayed on them. They have to eat it off of each other. If they get put through that vaccinator and then thrown on the truck and they're off, they are just jumbled around. The vaccine can fall off or it can sink into their fluff and it's inaccessible now. So giving the birds time in the right environment that's quiet, warm, letting them ingest it, that is key. So I'm going to go quickly through a few slides, just kind of looking at how the application could influence the cycling. So I think I've got 100 birds, and this is a barn. Forget about the brooding. This is just the production space. And let's look at a scenario where 90% of these birds receive the vaccine, and the birds that receive the vaccine have a green box around them, and those that do not have a green box have not been vaccinated. And so there's 10 birds on here that did not get a vaccine, and this would still be considered a good application. So if you were just like, imagine those birds are gone, but they've shed some parasites onto the floor, you can see we've got pretty even coverage of the parasites. These are the oocysts that come out a week later. They're all over the place. Great. So put those birds back in that situation. Okay, now really, like I said before, 90% received the first vaccine dose at the hatchery, and they're ready for the cycle now. So whoever was green before is now blue. So you can see that all these birds are blue because they've received the first vaccine, and now they're going to get vaccinated again. And then the birds who didn't receive the vaccine the first time, they're yellow. So they can handle this, but they may get a little bit more stressed because this is the first time they're seeing the cocci, and the pressure is a bit higher than what was given to the other birds at the hatchery. Okay, now let's look at it. If something were to go totally wrong and we get less than 50% of the birds get the vaccine at the hatchery. So again, now we've got 50 birds on here that do not have a box around them and 50 that do. And again, now the parasite burden in the barn is much more sporadic based on where they were when they went into the barn. And so when you put those birds back on that situation, we could now have three options. So we saw before the blue box, they received the vaccine the first time, and now they have exposure again. We also have those birds that didn't receive the vaccine the first time, but they are exposed to it here the second time. But what if there's the birds that did not get any vaccine the first time and they are not getting it here at the second exposure? So now we're going into the third cycle where the pressure is going to be building even higher, and the birds just are not ready for that. That's where I can see chaos ensuing. And that for A cerviline, it could be at 15 days. For Tenella, it could be 21 days. So these aren't true data. This is just concept to understand what it is that we need to be achieving when we're looking at uniform vaccine application. Hopefully everybody's still following and still awake. We're going to get into some field data here soon. Okay. We can also break our vaccines into either non-attenuated or attenuated, and it just means that they are slightly weaker than the attenuated ones. And this is because they've been chosen to have a shorter cycle in the bird. So like I said, those number of asexual replication cycles that happen inside the bird before they go to sexual differentiation, there could be 3 to 4. Well, they've got dialed it back to 2 to 3. So it's shorter now. So that's just the main difference. So it's going to be a bit easier on the bird, but it also means it's going to be slower for it to build its numbers. So there are absolutely advantages to both types of vaccines. So, okay, now let's get a little bit into monitoring coccidiosis in the field. A lot of the times vets will look for lesion scoring, and you can look at either birds that have died, or you can look at live birds to kind of get an idea. But this requires a little bit of expertise. It requires you to be present at the barn or birds brought in. I've been spending a lot of time over the last number of years looking at oocysts per gram. And this just involves taking fecal samples from throughout the barn. It gets submitted to the lab. In the lab, it's a fairly straightforward process. There's a little bit of expertise or know-how that's needed, but it's relatively quick. You're mixing it with salt water, a sieve, and then you're putting it under the microscope, and then the oocysts will float in salt water. And so then you can say, okay, well, what does oocysts per gram mean? It's essentially all the parasites that are coming out is likely to reflect the number of parasites that went in, and therefore the damage that occurred, and then so that's what's coming out. So that's why we're doing this. But I will put an asterisk to say that oocysts per gram is very tricky, and you can't always tie OPGs to, say, lesion score, or you can't tie it to lesion scores, and it's hard to really start to say OPGs determines mortality or determines lesion scoring, feed conversion, or weight gain. That's what I was trying to say. So where I really use oocysts per gram is to understand the cycling that's happening in the barn, that any areas of improvement for the management, that's where I'm using OPGs. And then it also can be used to monitor medicated flocks, and we'll talk on that too. So we can't take it at one point. We have to take a series of samples throughout the flock. And because it may change flock to flock where that pressure is going to be. Is it going to be at week 2, week 3, maybe week 4 if it's a medicated flock? So by getting a shedding pattern, it's going to be the most informative for us. Our day 7 count is a vaccinated flock is essentially dependent on the vaccine being applied. And then there's nothing really that can affect it other than the vaccine, unless you were to have a drug in the feed, then obviously but that's not going to happen often. Day 14 depends on what happened at the hatchery and then the management that was done after a week in the barn. And then same thing for day 21 or week 3. It's all building and good reinfection, good sporulation. And then finally, we should start to see, again, this is for a vaccinated flock, at 4 weeks, we should start to see the results of immunity developing. So we see vaccination success, and it's dependent on the cycling within the flock. So as part of my thesis, I took samples from medicated flocks, conventional program, vaccinated flocks, and we did it on a weekly basis. I would recommend, if you've got the resources, do it more frequently. Every 3 days would be awesome. But for us, this was the most logistically possible that we could do. And doing fresh feces is going to be different than if you do litter. But let's not dive into that today. And we did look at performance parameters, but you can find this information on Poultry Science. It was just published last fall. So go there if you want to find out more. And this is the same data taken straight from it. So our medicated flocks have quite variable OPG counts. So each of these lines is a different facility. As the birds age, you can see the pressure of the oocysts per gram is going up, but then comes back down depending on where you are. Nothing in here is controlled for hatchery, feed company, medication product, management. This is a catch-all. Whoever wants to participate, I would go to their barn. So nothing's really controlled for here. It's hard to pull anything out of this other than just trends. I'm going to keep this same axis the same, and I'm going to go to the vaccinated flocks, and you can see that there's a drastic difference between those. It's much higher from the previous slide on the medicated flocks compared to these ones. And we can see they all come up around 2 weeks of age. Some at 3 are peaking, but they're all coming back down, and most of the flocks and almost all had cocci present in the last week. So it's still there. So there's still a few birds trickling along, but again, which vaccine was used, we have it recorded, but there's nothing to be really pulled out of this. Which one's better? You can't tell from here. I tend to like the flocks that peak a bit sooner, but lower if you can. And that way the birds are just more protected as they go into, that 15 to 18 day old chicks where they're stressed with necrotic enteritis. So if you were to cluster that data and look at it side by side, you can see, well, the numbers aren't drastically higher for medicated versus vaccinated, but the pattern is quite distinct. So the other thing is, I started this talk off talking about all the different species. So when we look at OPGs, it's all of the species together. So when we look at the numbers that I showed, it shows different patterns, but what if we start to look at the different species that exist in there, it could be a totally different picture, and it could be even more complicated than this. This is hypothetical values, but there are technologies that are used today. You can either look at oocyst dimensions, but I don't fully rely on those. There's a number of labs that are working on molecular techniques to use DNA to say species A, B, C, and D, and then they'll quantify and identify those species for you. So, okay, so that's one concept, and it's being worked out. It is always tricky, but it's the future, and I think it's going to be really exciting when we can make it more affordable to do as a monitoring practice in our flocks. So why did we see so many flocks with challenges on the medicated side? And maybe not challenges, but at least why were they so much higher, right? The drugs should be working, they should be much lower than what we saw. And again, it probably comes back to some resistance developing. Again, not total bad resistance, just loss of effectiveness. And this isn't surprising because we have a series of publications from across the world showing different drugs that have developed resistance, parasites developing resistance to these drugs throughout the years. What's one way to work around this? There's a great tool by the Platinum Brooding Program, the Coccidiosis Prevention Planner, and this just allows you to map out what drugs you've been using throughout the year. So this is outdated, it's got A165, but you can see in your starter feed, you would use one drug, maybe a different drug in the grower and finisher. So you have in a year a complete picture of what your program's been, how much of each product you've used, and then you could just put that in your notes and say, here's the history from all the years past. Okay, now we can start to pick the drugs that we have not used in a while, or different programs to really suit the performance that you're desiring. So what can a producer do if they're having trouble? I've only got 10 slides left, so this won't be too much more. But say they want to stick on anticoccidial medications. They don't like the vaccine program, or they're not ready to put in extra oomph, I guess, into their management, and they want something. Well, the best answer that I've got for you is to still use the vaccine. So another publication that came out last fall that we worked on was restoring drug sensitivity, and we did this in Canada. So this concept is not new to the US. This publication came out in 2014. I think this was being used before that. But if you think about using drugs, so it says here chemotherapy, at the beginning of the year, and then in the US, you guys would clean out, but in Canada, we keep cleaning out at the end of every flock. But in the US, clean out, get rid of as much of the old litter as you can. And then we're gonna vaccinate for flocks 3 and 4 during the summer. And that's because it's the easiest to vaccinate at this time of year because we got good humidity, good outside environment. What they saw in this publication is that flocks 5 and 6 actually, even still using the same program as 1 and 2, but 5 and 6 had better performance. And that's because when we put these vaccines in, we're putting in drug sensitive parasites, as I mentioned earlier on. So we said, okay, how does this work in Canada? We clean the barn out every time. How can we do it? So we identified one of the farms with abnormally high OPG counts, and you would have saw that. I showed that data before. And so we said, okay, let's put 2 flocks on the vaccine, and then we'll go back to the drug and see exactly what happened. So you already saw most of these data, but this 2 million OPG in the first flock, and then it came down because they're on a different program, and then a different drug program, it came down even further. They vaccinated 2 flocks, and then you can see we went back to program number 1, and it's totally different. So if you look at this program top left and bottom right, totally different, even though it's on the same drug at the same farm. So whatever happened here was beneficial. But this isn't really perfect. We don't know what we did. We just changed the pattern. So we took samples from before and from after the vaccine going in, and we put it through what's called an anticoccidial sensitivity test. So here, there's a number of different ways that you can do this. We looked at 2 methods, including parasite biology, which is a low dose, and then looking at chicken performance, which is a high dose. And you can measure the reduction of oocyst output when drug is present, and that'll give you a category. We want to split these into categories rather than just giving it a number. And then in this high dose challenge, we looked at, body weight gain, feed conversion, mortality. And then with all of those, because it's hard, well, which one's the most important? We combined all of them into an anti-coccidiosis sensitivity index. And that is in the publication. And if anybody has any questions, I'd be happy to forward that to you later. And when we did this stuff, we always used the label dose, label requirements of the products in the mixed feed. So this sensitivity index was about, we gave it different scores based on what we perceived to be the most important. So I said average daily gain gets the most points, 50 points, because at the end of the day, we want our birds to grow. Next, we want our birds to grow with a good feed conversion, so it gets the next highest. Lesion scores, that is indicative of how well the drug is working, but it's not as important as the last two metrics. And then oocysts per gram, you can have high oocysts, but if everything else is fine, maybe that's starting to show the tendency of the drug starting to not work. So this is meant to say, okay, the perfect score would be a 1, and so 50 points for average daily gain and 30. So these are meant to add up to 100 if you have perfect on all of them. And so then you would then say, okay, well, if you're getting less than 30, you're resistant. If you are 30 to 70, you're partially sensitive. And then if you're above 70, that you're still using a strong drug. And so back to that that farm where we use the vaccine, we have the before the vaccine and the after, and you can see that we had a few drugs that were resistant, and then diclofenac was actually sensitive here. And then afterwards, we had 2 drugs that had gone up to be more sensitive. So we did prove that there was an improvement here. The Menensin just kind of stayed in the same category, and Zoline did as well, but this does prove that there is capacity for that. So to summarize, as we mentioned, coccidiosis, it's an artificial disease. We've got a lot of species that we're working with, and thankfully we found 3 more, so that just makes it more complicated. It's dependent on the cycling in the barn, and that's going to depend on whether you're looking at vaccinated flocks or medicated flocks. And we're always cautious about our bacterial infections. And we know that drug sensitivity, it can be restored by using the vaccine. So not all hope is lost if you want to continue using anti-coccidia medications. So with that, I would like to acknowledge a lot of the funding that went towards my research, including the University of Guelph, NSERC, OMAFRA, and the Ontario Veterinary College. Thank you so much to everybody at Platinum Brooding and the BC Poultry Symposium. It's an absolute honor to be speaking here. I've been looking at all the people who spoke or watching the previous presentations, and I'm truly honored to get the chance to speak at this. And I'm looking forward to the next presentation that's going to be on environmental impact that's coming up in about a month from now. So with that, I am going to take some questions. I see that there's a few in the Q&A chat box. So if you have some questions that you'd like to ask, type them in there and I will see if I can answer them. And one thing I don't think I have on here, so I apologize. My email, if you'd like to reach out to me, you can reach me at ryansnyder@grandvalley.com. So first name, last name @grandvalley.com. And I think I can get Stu to share that with people if they need to get in touch with me. Okay, so first question I've got, what time of day, if any, is better to treat coccidiosis in the chicken? I don't have an answer for that. That's a really good question. I will hazard a guess for you. I look at, when the infection is when you're first diagnosing the infection, and then if you're going to treat, it's likely through the water. So maybe when the lights first come on, if you have a lighting program where the lights are off, the birds are going to get up and all are going to go have a good drink. I would start there. Second question, can you prevent oocysts from adhering to the GI tract by water or feed additive, especially in early life, preventing GIT damage? Again, not a full answer because I will not claim to be a nutritionist. I have not seen any feed additive or water additive stop a parasite that is millions of years old from doing its thing. These things, I find the biology of the parasite is they are really effective at finding their way to the to the gut and finding their way in. I don't know of any way to stop them from finding the host cell. So I would say there is no way of stopping that. But is there a product to help the gut be stronger and tighter to fight off those infections? Maybe, but the actual mechanism of the parasite finding its way in is a bit different. And I don't think you can stop that. Next question. What % of flocks in Canada are vaccinated versus in meds for Cocci? I wish I had a true answer to that. My understanding is that the market share of raised without antibiotic is probably around 30 to 40% in Ontario at least. And those that are on that market are getting the vaccine. So I would say probably 30 to 40%. Yeah. Next question. In the test to restore sensitivity to anti-coccidials, do you think OPG variation could be caused by seasonality? And yes, absolutely, because you must have been sharp enough to see that I had some samples that were collected for shuttle program number 1 beforehand. It was in May and then again in December. I do think that could have an influence, but we typically would not see that drastic of a change within that barn. So there's some flocks that we had sampled that weren't included in there that I think you're right. It does have some influence, but the amount of shedding that we saw early on was totally different. So I'd have to go back and point that out again. And then as the second part of that question, and what performance FCR variation between flocks? That I'd have to dig up. I don't have a clear answer on that. The farmer, so when that farmer did have like the 2 million, it must have been ASERV there because they did not really have a whole lot of issues. They claimed that they said, yeah, like the barn seemed a little bit wetter, but it wasn't like birds were dropping. So that's why I think it was mostly ASERV line in that flock. And so maybe they did lose a bit of feed conversion, but I don't have that answer handy for you. So, and it's tough because, now we're looking at FCR between flocks. So it's a different hatch. It could be different nutrition in terms of the grains coming in. So it would be lovely to have, like a side-by-side direct comparison. So totally agree. That's it for the questions on here. Does anybody else have any more? I do see, actually, sorry, there's a chat. Why following a Cocci vaccine application, the OPG count in some barns peak at 2 weeks of age and in other barns at 3? Yeah, very good question. I do believe it may have to do with the vaccine. It could have to do with the vaccine application, the concentration of oocysts that are going to the birds. It could have to do with the success of the farmer with getting the sporulation just right during that first week. So, it may go up faster when you've got really good sporulation and really too condensed environment. Whereas if you leave the birds have access to the whole barn, it might be a slower crawl and it's going to take you 3 weeks to get to your peak and then later. I hope that answers the question. So yeah, to summarize on that, I would say it has to do with the vaccine application, and it could have to do with attenuated versus non-attenuated vaccines, and then the management in the barn. So I don't see any more questions coming in. I think I'm going to wrap up on time. So again, anybody has any questions, you can reach me at my email, ryansnyder@grandvalley.com. And it's been a pleasure presenting to everybody today, and I hope you all have a wonderful day.